This week, you're going to be covering chapters 1 and 3. In chapter 1, you're going to be learning about homeostasis, which is a really important concept that's going to weave through all of physiology. Here you have the textbook definition, but you can also think of homeostasis as the normal, most desirable conditions of body functioning that are necessary for our survival. Here are a number of parameters or variables. The variable is what's being regulated. These are all precisely controlled by homeostatic mechanisms. So, for example, normal body temperature is maintained at about 37 degrees Celsius. Heart rate varies, it's a pretty wide range between 60 and 120 beats per minute. Our blood pressure is maintained at about 120/80. Respiratory rate varies normally between 12 and 20 breaths per minute. pH, you'll learn about the pH of arterial blood ranges, a very narrow range, between 7.35 and 7.45. So 7.4 on average, plus or minus 0.05. The blood gases, oxygen, carbon dioxide, are also maintained within narrow limits. Nutrients like glucose ranges between 70 and 120 milligrams per deciliter. Ions like sodium Na, which ranges between 135 and 145. Units are milliequivalents per liter. It's really important that you include units with all of your values. A lot of these values can be found in the appendix at the back of the e-text, which is in Pearson. Appendix F has select blood and urine studies. So homeostasis is vital to the normal function of our cells. Most of these variable parameters are maintained by what's referred to as a negative feedback mechanism, which reverses a change. For example, we sweat to cool the body. In our homes, the air conditioner turns on when the room gets too hot. Very few variables in the body are controlled by what's referred to as a positive feedback mechanism, which amplifies a change. The deviation from normal gets greater. The classic example shown here is blood clotting. Childbirth is another example. When negative feedback mechanisms become overwhelmed, destructive positive feedback mechanisms can take over. This results in homeostatic imbalances. Heart disease is an example of a homeostatic imbalance. A lot of the negative feedback mechanisms are aided by medical therapy. So, for example, if you have high blood pressure, you take a hypertensive agent to lower the blood pressure. You'll learn about cell structure in chapter 3, looking at the structure of the plasma membrane. It's mostly lipid. Lipids are fats, and you know that fats can solidify. So interspersed among the phospholipids of the membrane are cholesterol molecules, which keeps the phospholipids from packing together and the membrane from solidifying. There are proteins interspersed throughout the phospholipid bilayer. Those proteins have a couple of functions, transport, for example, and you'll do a lab this week where you learn about cell transport mechanisms. So that's the next topic. Make sure you know the key differences between active versus passive transport mechanisms. One of the most probably difficult concepts in chapter 3 is the effect of varying tonicity on our cells. So if you look at this figure, actually, let me show you definitions of hypertonic, isotonic, hypotonic. The suffix tonic pertains to the solute concentration. Here you can see solute in brackets. The brackets indicate the concentration. And then if you look at the prefixes, hyper, means greater than. Iso means the same. Hypo means less than. If you have somebody with hypothermia, their body temperature is lower than normal. So these terms only make sense when you're comparing two or more solutions. This figure is showing the effect that solutions of varying tonicity have on our cells. If you look at this isotonic solution, if you were to place, this is a red blood cell, if you were to place one of our red blood cells in an isotonic solution, the double arrows here indicate that water is moving into and out of the cell at the same rate. So the size and shape of the cell is unaffected. One of the things that you need to know is our cells have a 0.9 sloppy 0.9% sodium chloride concentration, which is normal physiologic saline solution. They have a 5% glucose solution. So you might have seen a bag of 5% dextrose hung. A hypertonic solution then is anything that is greater than 0.9% sodium chloride. And you can see the effect on our cells is to draw water out of the cell, which results in the cell shriveling in a process referred to as crenation. The cell crenates, it loses its normal shape. The water normally inside the cell pushes against the membrane and retains the shape of the cell. This last part of the figure is showing the effect of a hypotonic solution. This would be distilled water, for example. Distilled water is pure water. There are no solutes in the water. And if you place a cell in a hypotonic solution, you can see that that water is going to move into the cell, causing it to bloat or swell. If it swells to the point of bursting, that's termed lysis. Specifically, when we're talking about the red blood cell, the term is hemolysis, hemo pertaining to blood. So it's important that you understand the effect that these solutions have on our cells, so you know when to give a hypertonic, hypotonic, isotonic, IV solution. If you just want to maintain normal hydration status of the body, you're going to give an isotonic solution. If you want to push fluids, if you have somebody who's severely dehydrated, you're going to give them a hypotonic. The last thing in chapter 3 deals with organelles, and something that I want you to think about as you're learning the function of each of the organelles is to predict what the physiological consequence of a missing or defective mitochondria is, for example. So if you know that it functions in producing ATP synthesis or production is going to decrease if you have missing or defective mitochondria, and therefore you're going to experience muscle weakness or muscle fatigue.